Uh, Hello everybody, this is Rochan here. Today I'll be giving a small presentation on the topic nucleus from the chapter Cell, the Unit of Life. So let us begin. So to begin with, firstly, I'm going to look at the overview of nucleus. So when I ask you what is nucleus, what is the typical definition of nucleus? It is very simple. It is the largest and distinct cell organelle. So it is a membrane bound cell organelle which is distinct, which is differentiable from other cell organelles. It is large enough to distinct. Okay. And it also contains the genetic material, basically the DNA, and also controls the metabolism of the cell. When I say nucleus controls the metabolism of the cell, how does the nucleus control metabolism of cell? This we will look at in the function of nucleus when we go in the end of this recording. Okay. Now coming to the discovery part. Who discovered nucleus? Nucleus was discovered by a scientist called Robert Brown. He's also contributed towards plant morphology and embryology. Okay, he has also given his studies in those morphology and embryology. Next, to go with the number of nucleus. This is very simple. What I mean by number of nucleus is how many nucleus can a cell have. So to go with the first condition, uninucleate condition means a cell has a single nucleus, which is seen in most of our animal cells like smooth muscle cells and etc. Okay, next condition is binucleate condition. Binucleate condition means a single cell has two nucleus in it. Where do we see these kind of cells? You would have heard about microsporangium in the plant kingdom. So what is microsporangium? Microsporangium is going to give rise to microspores, which is the male gametophyte of a plant, right? It is going to carry pollen grains. So this microsporangium has specialized cells called tapetal cells, and these cells are going to be binucleated in condition. Okay. Next, the third condition is multinucleate condition, where we see many nucleus in a single cell. That is called multinucleate condition. This condition is seen in cardiac muscle cells and some bone cells as well. Okay, so this is multinucleate condition. Next, anucleate condition, which means a cell completely lacks a nucleus. There is no nucleus in that cell. And this, you all will know this example, that is the erythrocytes in human cell. What do you mean by erythrocytes? They are red blood cells, red blood corpuscles, or what we call. Why is it that nucleus is not there in these kind of cells? It is to help in the functioning of RBC. So any modification is to enhance the efficiency of functionality. So that is why we see so many types of nucleus, in number, in location, and in shape wise. There is so much of modifications. It is because it is to improve the functionality of that particular cell. This is done. So what happens if RBC doesn't have a nucleus? It is going to get more space to carry oxygen. We all know RBC is the oxygen carrier in our body that is going to help in transportation of oxygen to all parts of the body. That is why erythrocytes in human cell is going to lack nucleus. This was in animals. Now, even in plants, we all know phloem is the food conducting tissue that is going to have a specialized cell called sieve tube. And those sieve tube cells are also going to be anucleate to help in the functionality of sieve tube cells. Okay, this was about overview about number of nucleus. Now we will just look at location of nucleus. Mostly in animal cells, we are going to find the nucleus in central part. What I mean by location of nucleus is in the cell, where is the nucleus present? So if I consider this as a cell, in most of the animals, it is going to be present in the center like this, small spherical like structure. Okay. Other few animal cells are also going to have basal nucleus. Where have we seen this basal nucleus? In structural organization in animals, we have seen that the columnar epithelial cells, they are pillar-like cells. They are going to have nucleus at their base. So base position, if nucleus is present, it is called basal arrangement of nucleus. Next, few animal cells are also having peripheral arrangement. So if you see in striated muscle cells, striated muscle tissue, out of that one cell, we are going to find some striations like this, and nucleus is going to be at the periphery. What do I mean by periphery? It means at the corner of the cell. Periphery means corner of the cell. And we all are aware that plant cells, those are going to have nucleus in periphery itself. Why? So imagine this as a plant cell. It is going to have a large central vacuole. So in all plants, we are going to find a large central vacuole, due to which nucleus is always at the corner of the cell like this. So that is about location of nucleus. Now we'll come to shape. Now we have seen most of the nucleus. Whenever we draw a nucleus, we draw it spherically, spear-like structure, correct? But there are modifications in this also. We in WBC, white blood cells, we have classification of WBC as well. So there is a type of WBC called monocyte, in which in monocytes you are going to have kidney-shaped nucleus or bean-shaped nucleus. Similarly, in another type of WBC called granulocytes, you are going to have lobe-shaped nucleus. So why this modification here again? It is to help in the function of that particular cell. Okay, that is the overview of nucleus regarding discovery, definition, number, location, and shape. Now let us move on to structure of nucleus. So nucleus is going to be, as I told earlier, is going to be a membrane-bound cell organelle. First point to note here is, it is not a single membrane bound, it is a double membrane bound structure. So you can see in this figure, the nucleus is having two membranes, namely inner nuclear membrane, which is inside one, and outer nuclear membrane. So it is going to be having two membrane, double membrane bound structure. With that, you can see small pores here, pore region, correct? We can see small pores. So that is also going to be present in nuclear membrane. Next, coming to the next point, bilipid layer. What do I mean by bilipid layer? So if you take a section, or if you just magnify the part of membrane, either you take if this is the nuclear membrane, this is going to be the inner membrane, this is going to be the outer membrane. So either one of the nuclear membrane, if you just magnify either one of the nuclear membrane, what are you going to find? You are going to find a structure like this. And this is called fluid mosaic model or fluid mosaic membrane. Where have we seen this? This was used to explain the structure of a cell membrane. So this was proposed by Singer and Nicholson. So any membrane for that matter, be it cell membrane, nuclear membrane, or any other membrane, is going to be mainly composed of this bilipid layer itself, with some modification in proteins and all those things. But every membrane is going to be a fluid mosaic membrane. So even the nuclear membrane is going to be a fluid mosaic membrane with a bilipid layer, extrinsic, intrinsic proteins, and all those things present there. Okay, this is about what we see if we magnify a nuclear membrane. Next, similarly, we saw that nuclear membrane has pores. Why do we need pores? Obviously, for exchange of materials to happen, nucleus cannot be isolated. It will require materials from the cytoplasm of the cell. It has to be giving out some of its content, whatever is not required. That is why we need some kind of passage. That is why the pores are present. So what now, pores are just not present. They're going to guarded by special proteins called nuclear pore complex. What is this nuclear pore complex? Let me just show. If you see here, I have magnified the pore region. Sorry. So this pore region, if you just magnify, you are going to find these kind of structures. These what we see here, that is called nuclear pore complex. What is its job? So if any material has to enter into the nucleus, it is going to enter through the pore itself. If anything has to get out of the nucleus, if anything has to exit the nucleus, that also is going to be by this pore itself. So what this nuclear pore complex does, it acts like a doorway to the passage. So for that pore, it acts like a doorway. So if any enzyme is trying to come in, the nucleus is going to tell the nuclear pore complex. When I say nucleus is going to tell the nuclear pore complex, the genes, the DNA of the nucleus is going to tell the nuclear pore complex whether to open the door or not. The enzyme can be harmful to, it may be needful also. So if the enzyme is needed, if the nucleus needs that enzyme, then it is going to tell the nuclear pore complex to open the door and the enzyme is going to get in. Any material, not only enzyme, it can be mRNA, tRNA, other proteins, etc. So similarly, the genetic material, we saw that DNA is present here, right? Chromatin. So the chromatin also should not escape out. Okay, so to prevent that, these doorways are present, which are called nuclear pore complex. So what can we say that because of this, the nucleus is selectively permeable? What do I mean by selectively permeable? It means it is not going to allow every material to come in or everything to go out. Then it is going to be a big problem. So it is going to select the materials whichever it requires, and it is going to allow and exit uh, uh, the materials. Okay, so it is going to control the movement of molecules. So that is about nuclear pore and nuclear pore complex. With this, an additional information, or it is commonly observed that always nucleus, not always, most of the times, nucleus is going to be associated with endoplasmic reticulum, these kind of structures. And in that, especially RER, rough endoplasmic reticulum. Why do we see always nucleus is endoplasmic reticulum is technically not a part of nucleus, but still most of the times we see that it is attached to the nuclear membrane, outer nuclear membrane. Why is that so? Very simple. We have seen in the functions of RER that RER is the one which is going to produce the nuclear membrane. So this membrane is formed by RER only. If there is no RER, then no nuclear membrane. So we can see here also the functions are very much related. So because of RER, because the function of RER is to produce nuclear membrane, it is going to produce, and that is why we can see the structures are also linked. So that is why we see most of the times RER is going to be attached to the nuclear membrane. Next, the second region or the structure which we find in nucleus is perinuclear space. Very simple. In this diagram, you can see between the inner membrane and the outer membrane, you have this gap. This gap is called perinuclear space. So the region between inner and outer nuclear membrane is going to be called perinuclear space. That's all. Okay. Next, coming to the fluid part of nucleus or the matrix part of nucleus, that is nucleoplasm. It has two other alternative names. We can call it nuclear sap or karyolymph. Karyo means nucleus, lymph means the fluid part. Okay, so what is this nucleoplasm? It is just like cytoplasm, So cyto means cell, plasma means fluid. So it is the fluid part of the cell. Similarly, nucleoplasm is the fluid part of the nucleus. That's all. What we can see in this nucleoplasm, we can see chromatin and not nucleoplasm. We can see chromatin and nucleolus suspended. Okay, nucleolus suspended in the uh, nucleolus suspended in the nucleoplasm, right? Is it only this much? No, we can see other several minerals which the nucleus will need for its nourishment, like magnesium, potassium, calcium, and other minerals we are going to see. What is the main function of the nucleoplasm? Just like how cytoplasm gives a shape to the cell. Similarly, nucleoplasm is going to give shape to the nucleus itself. Okay, so that is about nuclear membrane, perinuclear space, and nucleoplasm. So this we completed the outermost part and the fluid part. Next, let us look at the two important structures, that is nucleolus and chromatin. What is nucleolus? It is very simple to define. It is a small dark spherical structure. Just like nucleus, you can call it a mini nucleus present and suspended inside the nucleoplasm. So very important point to notice, it is, one second. Very important point to notice that it is a non-membrane bound structure. Nucleolus is a non-membrane bound structure. So what is it going to do? What is the function of nucleolus? It is related with the formation of ribosome and ribosomal subunits. Okay, we all know ribosome. The function of ribosome is protein synthesis. Similarly, ribosomal subunits is nothing but one RNA plus one proteins put together is going to give one ribosomal subunit. So what kind of RNA? We have many kinds of RNA. rRNA, ribosomal RNA plus proteins put together is going to be called ribosomal subunit. Even its function is going to be protein synthesis itself. So not directly, indirectly, the nucleolus is related to the protein synthesis of a cell. Okay, that is its main function. Now, where is this nucleolus going to be formed from? Where is it derived from? It is going to be derived from the DNA. We saw the chromatin is suspended in the nucleus, correct? Chromatin is here. So if you see, one part of this chromatin is going to be, one part of the DNA is going to have a region called NOR, nucleolar organizer region. And this region is the one which produces nucleolus. So the DNA, the chromatin, inside the chromatin thread, DNA is going to be present, and in that DNA part is called NOR region, or just NOR, nucleolar organizer region, and from that region, nucleolus is going to be formed. That is about formation of nucleolus. When this NOR is producing nucleolus, it is not necessary that it produce only one nucleolus. A nucleus can have more than one nucleolus. So one is here, one can be, another one can be here, another one can be here, and there can be many nucleolus. So I have written one sentence here. Number of nucleoli per cell varies, per nucleus varies. Hence ribosome production can vary. Hence protein synthesis of a cell also varies. What do I mean? So if a cell has many nucleolus, obviously it means more number of ribosome and ribosomal subunits are produced. If more ribosomes are produced, means more protein synthesis, correct? So that is what I mean. So if a cell has, say, 4 nucleolus, and if another cell has only 1 nucleolus, the cell having the nucleus or the cell having 4 nucleolus can produce more protein or can synthesize more protein than the nucleus having only one nucleolus. That is what the last line means. So that is about variation of number of nucleoli per cell or per nucleus. Okay, and this was discovered, Nucleolus was discovered by Fontana and it was named by Bowman. What we study as Bowman's capsule, the same scientist, Bowman, discovered named Nucleolus. Discovery was made by Fontana. This was about Nucleolus. It was about Nucleolus. Next, we'll come to the most important part of nucleus, that is the chromatin. What is chromatin? We all know it is the genetic material. It contains DNA. When I say it contains DNA, does it contain only DNA? No, it is going to have DNA as 40%, that's all. So if you see in the diagram, this is the chromatin. From this set of chromatin threads, I repeat, chromatin is never going to be present as a single thread. It is always going to be grouped. Many thousands of threads are going to be together. So if I take off one thread like this, and if I magnify this thread, so if this is one thread and if I have magnified this, I'm going to find this is the DNA strand. Okay. With that, I'm going to find structures like this. So these round structures are bounded by DNA thread. What are these round structures? These are going to be proteins. When I say proteins, can it be any protein? No. It has 60% of proteins, mainly called histone protein. So it is going to be mainly called histone protein. So 40% DNA and 60% of histone protein. There are going to be some proteins called non-histones also, but mainly it is going to have histone proteins. Okay, so it is highly extended network of threads. It is never singly present. So what is going to happen during cell division? When a cell is dividing, these thread-like structures is going to form some structure like this. What we call this is chromosome. This is called chromosome. So chromatin is going to form chromosome during cell division. That is about chromatin. And it was discovered by Walter Fleming. So chromatin helps in the genetic, it has genetic material which helps in transmission of traits, characters from one generation to another generation. So this is about chromatin. Next, let us quickly look at, we are done with the structure of nucleus. Now, this is how it looks. We have discussed everything. ER, nucleolus, chromatin, nucleoplasm, nuclear pore, nuclear envelope, and also ribosome is going to be present on RER, correct? And nuclear pore complex, if you just magnify nuclear pore region, we saw nuclear pore complex, right? This is how it is going to look like. Nuclear pore complex. Next, so this is the structure of nucleus, right? Next, let us quickly look at functions of nucleus. Very simple. It contains the genetic information in form of chromatin, which helps in transmission of genetic information from one generation to another generation, which means it controls the hereditary character of an organism, right? Second, if you remember in the beginning, the definition of nucleus, I told it controls the metabolism of cell. And I told we'll look into it in the functions of nucleus. What do I mean when it regulates the metabolism of cell? First of all, metabolism means sum total of all chemical reactions happening inside the body. And all metabolic reactions are enzyme catalyzed. So you need enzymes for all metabolic reactions. And now quality and quantity of enzymes is going to be by nucleus itself. So how much enzyme should be produced? What is the quality of enzyme is taken care by the DNA of nucleus. So if more enzyme and more quality and quantity wise, if it is produced, then more metabolism can happen. Okay, that is about regulation of metabolism. It maintains the structure and functions. So as I told you, if anything has to enter into the nucleus, then the nucleus itself is responsible. Or say, for cell division to happen, we all know the first step is karyokinesis. Karyon means nucleus, kinesis means division. So if karyokinesis has to start, who is going to initiate the karyokinesis? It is the DNA of nucleus itself. The nucleus is going to tell, okay, cell divide. Then only it is going to start dividing. That is about maintaining the structure and functions. Now it controls the cell division and growth. So if a new cell is formed after cell division, it is going to give a new set of instructions to the new cell. Just like if a new factory is set up and to the employees, the chairman or the CEO is going to give a set of instructions, do's and don'ts. They must follow that. If they don't follow, they're going to be fired from the factory. They're going to be fired from the company. The same way, every instruction given to the cell is given by the nucleus itself, and the cell must follow. If it is not going to follow, then there's going to be a problem there. Okay, so that is how it controls the cell division and growth. So this was the overview of nucleus. I hope it was clear. Thank you so much.